According to the World Health Organization, cancer is the second most common leading cause of death and responsible for estimated 9.6 million of deaths in 2018. Globally, 1 out of 6 deaths is due to cancer. Although lung cancer is the only second most common cancer in both men and women, it's still leading the cause of the disease. According to the Diagnostic, the frequency of diagnosis on early stages, when curative treatment is possible, is very low comparing to advanced stages. This is because early stages are asymptomatic. If we look for the frequency of diagnosis on the late stages, advanced stages, we see that percentage are more higher because the tumor has metastasized. If we look for the current diagnostic as biopsy in chest radiography, it is not effective on early stages because it cannot detect the tumor until it's small. I believe that cancer burden can be reduced through its early diagnosis. The aim of my project is to evaluate if the breath can be used for early stages of lung cancer diagnostic. My solution is in this project. My project proposes a non-invasive way of lung cancer detection on early stages through several steps. Each of them corresponds different tasks. However, I'd like to detect those through extracellular vesicles, exosomes, that collected from breath. Exosomes secreted from cells actively participate in cellular communication, transmitting signals by protein, lipids, sugar, and RNAs. Exosomes secreted from lung cancer will contain biomarkers and therefore can be used for diagnosis. For that, for diagnostic purposes, we do need a high purified quality exosomes. For that, I have compared two methods of exosome isolation, ultracentrifugation and size exclusion chromatography. And I use electronic microscopy to examine the morphology of exosomes. Exosomes has been validated through gold nanoparticle conjugated antibodies that targeted specific exosomal marker. My next step will be to conduct protein studies through mass spectroscopy, where I will get the information about the proteins. Then I will conduct a library of biomarkers from lung cancer and healthy cells that will help me to understand the difference. And then I will compare those proteins from lung cancer and healthy cells with proteins collected from breath exosomes. I believe that my research can break the walls of early detection lung cancer through non-invasive steps.